Boa tarde a todos, sejam bem-vindos a mais um webinar da RBCC. Esses nossos encontros quinzenais têm sido muito produtivos e eu tenho certeza de que temos aprendido muito sobre cromossomos e genoma e tem sido possível reduzir em muito, né, a distância entre nós e possibilitado a gente ter contato com pesquisadores de grande renome na área. E não diferente disso, na nossa palestra de hoje, a gente tem a honra de receber uma importante referência para todos que estudam cromossomos sexuais e sistemas de determinação sexual, que é a doutora Nicole Valenzuela. Ela tem gerado dados e contribuições científicas muito grandes nessa área de estudo e nós estamos muito felizes em podermos contar com a sua presença aqui no nosso canal. E para nos ajudar a conduzir esses trabalhos de hoje, eu conto com a participação novamente da doutora Marta Svartman, da Universidade Federal de Minas Gerais, e também agradeço o convite feito pelo professor Marcelo Vicari para a nossa palestrante de hoje. Nossa interação será, como sempre, através do chat, então as perguntas e comentários poderão ser publicados no chat, e nós iremos transmiti-las aos nossos palestrantes e conduziremos, então, a nossa discussão depois disso. Então, vamos iniciar a nossa terceira live da RBCC Webinars. Today, we have a privilege to meeting Dr. Nicole Valenzuela, an important reference on sex chromosome studies from Iowa University, to talk about your experience on sex chromosomes and genomic context of the sex determination. Dr. Valenzuela is professor from the Department of Ecology, Evolution, and Organismal Biology from Iowa University, and lead head of Evolutionary and Ecological Genomics Lab. Your lab are interested in studying how ecology affects the structure, function, and evolution of the genome, and its role on the development and evolution of complex phenotypes. Thank you so much for your presence here, Dr. Nicole. I said before, but it's a great privilege for us to learn about your research. I wish a great lecture for everyone, and please welcome Dr. Valenzuela.
Thank you, Daniel, for that introduction, and thank you for all for being here and joining me today, even in long distance, so that I can share with you the work that we have been doing in the lab with respect to the evolution of sex chromosomes in turtles in particular. So I think to share my screen, I need to go to present now. Let's see, we did this before, your entire screen. Then I'll do that, I'll share. Then I need to do that. That's
it's working. It's working.
Perfect.
Perfect.
Thank you. So I'm originally from Colombia, and this picture is a turtle that is dear to my heart, Porognimis expansa, that lives in Brazil also. Is the famous arrau turtle from the Amazon basin. And I started working on turtles, trying to understand sex determination in this particular turtle, the arrau turtle. But later, trying to understand the temperature-dependent sex determination. Today, I'm going to talk into a whole different area that relates to the evolution of turtle sex chromosomes and their dosage compensation. And first, before I get to dive into the main content, I wanted to acknowledge students, postdocs, and collaborators that have contributed to the work that I'll present today to you. I'll start by telling you a little bit about what we know of turtle sex chromosomes, how we have identified them, a little bit of what they know of their content and their evolution, and then I'll get to the issue of dosage compensation. First, a general overview, and then telling you how turtles are contributing to our understanding of dosage compensation. So let's start by guiding you here. For those who are not familiar, turtles have two main types of sex determination. Most of them that are here in this is a prune phylogeny, so it contains the turtles for which we know the sex determining mechanism. In red, we have temperature-dependent sex determination, or TSD. That seems to be in this phylogenetic analysis and is the ancestral condition. So the grandmother of all turtles had this temperature-dependent mechanism. But from that ancestral TSD, we have genotypic sex determination, which involves sex chromosomes, that evolves at least 5 times in these lineages colored in blue. Different parts of that phylogeny. But the prevalent mechanism that we know in turtles is the temperature dependent. Now, these 5 evolutionary events that gave rise to sex chromosomes is what is the sort of the central topic of my talk today. This is the same phylogeny, but in a different, shown differently, and showing in particular that we know that some of these sex chromosomes in turtles are XY, similar to humans. Some are ZW, similar to birds. And the one important aspect from a cytogenetics point of view is that these sex chromosomes in turtles vary in their shape and in morphology. So sometimes they are large chromosomes, like in these ones here that are macro chromosomes, several of these cases. But there's also cases of tiny dot chromosomes that serve as have that function of sex chromosomes in some lineages, like in these turtles here, these soft-shell turtles here. There's also some reports of chromosomes in turtles that have been debunked from, for instance, in this lineage that in the 1975 was reported as having another case of ZW chromosomes, and that was apparently an error in karyotyping that was corrected this past year in 2020. So we, in our lab, we have used a comparative genome hybridization techniques that probably you are all familiar with, where we paint the male and the female DNA with 2 different colors, and we use those paints to hybridize in metaphase chromosomes of males and females. And whenever there's a chromosome that's specific to only one sex, or a region on a chromosome that's specific to one sex or the other, we will see either only red or only green. And that's how we detect sex chromosomes. And this is the example of Glyptemis insculpta, one of the turtles. This is a North American turtle that we discovered the sex chromosome system is an XY system. And the male-specific regions in green here are found in some areas, in some bands in the Y chromosome, not in the entire Y chromosome in these males. We also use techniques like back fish, so in situ fluorescent in situ hybridization of back clones. We do have a back library of a turtle, a painted turtle, that is also a different North American turtle that has TSD, but we have a back clone library to draw from. And so we have different markers that we painted across different species. But in the case of this Glyptemis insculpta, we were able to look at the order of these markers in the X and the Y. And doing that, we discovered that the sex chromosomes of Glyptemis insculpta are homologous, correspond to the chromosome 4, the 4th largest chromosome in the painted turtle. That's the one with the back clone library. And that the order has changed, so we were able to identify an inversion that occurred during the evolution of sex chromosomes in glyptemies from the ancestral condition that is represented by the painted turtle, and also some different inversions that are that occurred during the evolution of the X and the Y after the evolution of the sex chromosomes. So these are the types of things that we are looking for in the lab, trying to understand the evolution of the sex chromosomes. These glyptemies, sex chromosome, this is the turtle in its natural habitat. They represent the youngest system of sex chromosomes that we know in turtles. And this is, again, the phylogeny of turtles with known sex-determining mechanisms. The color of the branches here correspond to the number of chromosomes in the karyotype of these turtles. And here is the lineage of Glyptenis insculpta of this animal, and they split from relatives that have TSD around between 20 or 25 million years ago. And it is in that genus, in Glyptemis, this is the one from the XY from Glyptemis insculpta. We know that a congener, a relative Glyptemis muhlenbergii, has also the same XY system by using molecular markers. So this one was, we detected the sex chromosomes by PCR, the polymerase chain reaction. And with that, we know that the genus Glyptemis acquires sex chromosomes. So it should have occurred at the split of that lineage from its relatives. So we're talking about a sex chromosome system that is 20, 25 million years old. And that for turtles is the youngest, because other systems that we have identified are older than that. Some are as old as 90 million years or more, or perhaps 150 million years. Now, interestingly, this, just to give you a little bit of an interest in ecological context, these sex chromosomes evolve in turtles multiple times, those 5 times here with the red stars, at times when the global temperature of the planet was hitting peaks. So the global temperature was at a peak in terms of global climate. So there's a moment where it's either at the peak or in transitions of climate change. And in those lineages, so that's an interesting, that there may be a correlation of climate change with the evolution of sex chromosomes. Now we also see that 2 potential reversals from sex chromosome systems back to temperature-dependent sex determination in this lineage with the blue dots that are accompanied by an accelerated evolution, molecular evolution in this genome. So that happens in every time there's a transition in sex determination, there is an acceleration in the molecular evolution of these genomes, but it accelerates even more during these potential sex reversals back to TSD. And there is a correlation between the transitions in sex determination and the rate of evolution of the number of chromosomes. So the karyotypes are reshuffling as these changes in sex determination occur. This is another turtle, Apollonis spinifera, spiny soft-shell turtle, North American, that we discovered the sex chromosomes almost 8 years ago. And this one is one of the cases of microsex chromosomes. So here are the metaphases, and you notice that the sex chromosomes here is this small chromosome compared to the large macros. So it's a relatively smaller pair. The W is larger than the Z because it has accumulated repetitive sequences in this region where the female-specific signal was also detected by comparative genome hybridization. The red here is in situ hybridization with repetitive sequences. That that system of chromosomes, the ZW, was also has undergone another change. One gene that is important for sex determination, the gene SF1, which is important for the development of testes in vertebrates in general, translocated from an autosome in the ancestor to the Z and W, to the sex chromosomes of this Apollon spinifera. And this is the Z or W chromosome that has a hybridization signal of a back clone containing the SF1 gene. And this is one of several genes that move about in the chromosomes of different turtles. This was work by a former student in the lab who looked at 10 different genes that are important for sex determination, either male determining genes or female determining genes, or genes that are important in the formation of testes and ovaries. And she looked at the chromosomal position of these genes in 6 turtles that have a TSD. In orange here are all of these species that have TSD, and in purple are the GSD, the species with 6 chromosomes. And what she found is that some of these genes remain, have not changed chromosomes. So they, for instance, are found in the largest chromosome across all turtles, except for this one, Hemiduram aquarii, that these genes are in the 3rd largest chromosome, but color-coded here in these cells, in this table, are chromosomes that are homologous. So even though it's the first one in most turtles and the 3rd largest in Hemiduram aquarii, they are homologous chromosomes. We know this through chromosome painting. These other genes, these 2 are in chromosome 3 of all these turtles, except in Hemiduram aquarii, they are in the largest. But they are, again, a homologous chromosome. And so on for these other ones. But there was the SF1 gene I just mentioned here, and FOXL2, another gene that is important for female, so it's an ovary specific gene, whereas SF1 is more of a testis development gene, that they are autosomal in other turtles, but they were translocated to the 6 chromosomes in Apollon spinifera. This is ASSP. Now, these 3 genes here, though they are in the same chromosome across all turtles, and it is a homologous chromosome, they have changed relative position. So this is the graph here in the bottom, is the phylogeny again of these turtles, and the position of those 3 genes in different colors, and how in some animals, in some species, they have undergone an inversion. In that case, in this case, in some cases, the spacing between them has changed between turtles. And there's another inversion here, a rearrangement of those genes in Hemiduram aquarii. So they have suffered changes in the position, but these changes do not correlate with changes in sex determination, because we see modifications, we see rearrangements that are found across TSD species, or some that are found that are shared between the order of the genes is shared between GSD and TSD species. So there's no change here, no rearrangements that is correlated with sex determination. The only one is the translocation of FOXL2 and SF1 to the 6 chromosomes of Apollon turtles. Okay. Now, in Chelidae, this is another family of turtles in the suborder of turtles where the Arau turtle belongs, the side necks, the turtles that can hide their head by twisting their neck to the side. And we discover first in this Australian turtle, Chelodina longicolis, the first micro-sex chromosome system in turtles. This was work in collaboration with our friends, our colleagues in Australia. Jennifer Marshall-Graves' lab and Tariq Essas did a lot of the work. And that's when I went there and learned how to do chromosomal work, but that was at Jenny's lab. Then this work was followed up by another work from a student of mine, also in collaboration with a group in Australia, with Hemidura macquarieae. That's where we found these two are in the same family, Chelidae. They are closely related turtles, but the sex chromosome system in Hemidura, even though it's XY, is a different XY. This time, the male-specific signal in red is found only at the tip of the Y chromosome, and is the fourth largest chromosome, unlike these tiny dot chromosomes in Chelodyna longicolis. And the same student of mine, Lindsay, later, that did the other work, did also discovered that a relative Hemidura, also Hemidura subglobosa this time, has the same large macrochromosome system as macquarieae, with the male-specific signal at the tip of the Y chromosome. So this and that led to the question as to how this large XY system evolved, because we wonder whether these tiny dot chromosomes were homologous to the XY in Hemidura, and that the Y could it have translocated to the tip of a chromosome and form this large Y. And that's what we basically found. So what we did, we revisited some data from the Australians who had some other chromosome painting, and found that the XY of Chelodyna longicolis is the same, is homologous to this. It paints the Y of Hemidura turtles. And so by revisiting the data and what we found is that if we look at the tree of turtles here, again, the phylogenetic relationships and the chromosome number of these sites. So these are the cryptodierans. These all are the ones, the side necks, the plerodierans, that to which Hemidura macquarieae belongs. It has this large XY system. Hemidura subglobosa has that same large XY chromosome system. Longicolis has this tiny dot one. And from what it seems to have happened is that the ancestor here of these Kelly turtles had a system, when it evolved GSD, had this tiny microchromosome, XY macrochromosome system. And the Y chromosome translocated to a pair of autosomes, forming a neo-X and neo-Y system that we see in Hemidura macquarieae. The X, because if we revisited the painting data from the Australians, the X chromosome seems to have disappeared. And so Chelodyna longicolis retain this ancestral condition of the tiny chromosomes, whereas in this Hemidura lineage, at least at the base of the Hemidura, we see the translocation and the evolution of this large neo-XY. However, there's new data from another group in Prague, from Lucas Krakowil and his team, and they found that Elseia Nova Guinea has exactly the same large XY system. So instead of that translocation happening here around 40 million years ago, or 35, it must have occurred earlier at the split of this entire lineage of Hemidura and Elseia. And so that will correspond with the reduction of chromosomes from 52 to 50 that happens in this lineage. Okay. Interestingly, also in the evolution of sex chromosomes in turtles is that there's a big difference of what's happening in the morphological evolution in different lineages. In example here, we have once more the phylogeny of turtles, Gray's TSD. In color, we have XY in red, and ZW systems in blue, although actually this is the one that is now known to have been an error. But the point I'm trying to make here is pay attention down here in the Chelidae, compare that to the soft-shell turtles, Trionychidae, these two families. The sex chromosome systems in both of these are about the same age, around 95 million years old. And yet in the soft shells, this ZW system hasn't changed much in morphology. We have here the ZW with a microchromosome, this expanded region of the W that is found in Pelodiscus sinensis and in Apollonius spinifera soft shells. And instead in Chelidae, we see the evolution, presumably once of XY at the base, at the split of Chelidae. And we have that Epidural aquarii have this neo XY chromosome system with a large macrochromosome. Chelodina has this tiny system, and Acanthochelis radiolata here, which is this lineage right there, has a macrochromosome, but this time the Y has degenerated, has decreased in size over time. So a lot of morphological evolution in the sex chromosomes in Chelidae, and yet very much stasis of morphology in the sex chromosomes of soft-shell turtles that are about the same age. From the sex chromosome systems for which in turtles for which we have genomic information, so information about the content of these chromosomes, we can trace these chromosomes to different chromosomes found in birds. So we know the homology of, say, the Glyptonis insculpta here. The sex chromosomes are homologous to chromosome 4 of painted turtle, as I mentioned earlier. They are homologous to the XY of Sibieronchila crassicolis, another turtle that evolves sex chromosomes utilizing the same ancestral autosome. These are homologous to chicken 5, but did so independently. So this is a case of convergent evolution of the reuse of the same autosome that becomes an XY sex chromosome system in Glyptonis and in Sibieronchila, but independently from each other. In Staurotypus triprocatus, we have, this is a Mexican turtle that has XY sex chromosomes homologous to the sex chromosomes of birds, the Z and W of chicken. And these soft-shell turtles, the Pelodiscus and Apolloni, have a Z chromosome that is homologous to chicken 15. Now there's this other one, chicken 26, that contributed to the short arm of the Glyptonis sex chromosome. When we look at this, then we see is independent recruitment of autosomes as sex chromosomes in turtles. So not homologous sex chromosome systems, except for the case here of Glyptonis and Sibieronchila. But if we look at the chicken chromosomes that are homologous to the turtle sex chromosomes, we notice that they share a deep homology with amphibians, with Xenopus tropicalis chromosome 1. So color-coded in Xenopus 1 are the blocks of this chromosome that are homologous and gave rise to these different sex chromosomes in turtles. So this arm of the Glyptonis, the short arm that is 26, is found here in Xenopus 1. This area that contains the MRT1, an important male determining gene, and the sex determining gene in birds, is found in this block of Xenopus, plus this other one also contributes to chicken Z. This part here does have homology with the human X, and these other leaves are Z chromosome, and the green block gives rise to Anolis carolinensis leaves are X chromosome and the Z chromosome of these softshell turtles. So that raises the question as to whether there was an ancestral protosex chromosome, either this big one, chromosome 1 in Xenopus, or even in more ancestral one that might even have incorporated what is Xenopus 5, which is the homologous to chicken 5 and these two sex chromosome systems in turtles. If there was one huge chromosome ancestral that for some reason its content, other properties, makes it good at sex, makes it good to become a sex chromosome. So that's a question that warrants research in the future. Now, what about the business of dosage compensation? So let's jump to that. So in general, the canonical classic model of sex chromosome evolution goes something like this: that in some ancestor there is a pair of autosomes that acquires a sex-determining factor. In the example illustrated in this graph is a testis-determining factor, so it's a male-determining gene. And once that mutation arises and is found in males, you have here a proto-Y chromosome. The companion will be the proto-X, and the two chromosomes of the pair found in females will be proto-X. Over time, if that same proto-Y chromosome also starts accumulating male beneficial genes that are detrimental to females, so there's sexual antagonism in these genes, then keeping these two genes in a block that doesn't recombine, that doesn't break down this nice adaptive combination, is favored by natural selection. So therefore, there will be the reduction of recombination favored by natural selection through things such as inversion, chromosomal inversions. And that reduced recombination permits the accumulation of mutations that are mildly deleterious, and that accumulation starts the process of degradation of the Y chromosome that eventually result in the degradation of the loss of function of genes and eventually loss of gene content, just pieces of DNA, so that these chromosomes could shrink in size. And that would happen, so cessation or lower recombination, between the X and the Y, except for an area that is pseudo-autosomal, where recombination remains intact and permits also the assortment of the chromosomes during cell division. But when that happens, that means that the genes found in this area of the Y chromosome lost function or were lost completely. So now males will have genes in the X that are missing from the Y, but the females will have two copies of those genes. And that then brings us to the need for dosage compensation, because when here in this first row we have, if there's no dosage compensation and we have an XY system where males have a degenerate Y, they're missing genes that are found in the X. Therefore, they have only one copy of these X genes and the females have two copies. So there is a greater number of copies in the female. So the balance of dosage between the sexes is a female bias. And then these in the males could be detrimental if the products from these X genes need to be in the same amount as in balance with products from the genes in the autosomes, because in the males, the autosomes have more dosage and are producing more proteins that the only X wouldn't be able to, unless there's a mechanism of dosage compensation that will balance this difference in the dosage between the male X, single X, and the autosomes, and that will lead to the balance between the two sexes. How this is done varies by lineage. In mammals, is done by first the inactivation of the female, one of the female X's, and then the doubling in transcription of the single X in the male and the only active X in the female, and that brings to parity sexes and autosomes and sex chromosomes. In fruit flies, is simpler, just ramp up the transcription of the only X in males, and that has the same result in a single step. And in C. elegans is a little bit different, but similar to steps compared to mammals. All of those systems are chromosome-wide with some SCAPI genes, and and that was thought to be the way that dosage compensation happened, when first that was the only dosage compensating mechanisms that were known. But then people looked at chicken, people look at mouse, and they found no global dosage compensation, but something that happens gene by gene that is able to balance this difference in the dosage between the males and females and the autosomes. So it happens locally, gene by gene. And more systems were then discovered that are neither one nor the other. So the diversity of sex chromosome dosage compensation is now more than we knew early on. The evolution is still obscure, and here is where turtles are a good model to study the evolution of sex chromosomes, because we have systems that are XY, systems that are ZW, that are independently evolved. So we have recurrent evolutionary events that gave rise to sex chromosomes, so we can test whether dosage compensation exists in turtles, whether it's global or local, whether it matters if the chromosomes are younger or older, bigger or smaller, and so on. So to start with that, that's an ongoing project in the lab. And we're looking, I'm going to show you data from Glyptemis and Apolloni, starting with Apolloni. And the first thing that we had to do was to figure out the content of these ZW chromosomes. And we did that by sequencing a male and a female, low coverage, and then map these to a reference genome. And here is when you map the reads of the males here in red and female in black, you see that as you're mapping these reads to the scaffolds of these chromosomes, when you have an autosome here in this bottom left, the number of reads from autosomes in males and females should be pretty much the same. So if you map the abundance of these reads across the scaffold, you see that the male and female reads pretty much overlap with each other. That's what you should see in autosomes. For the case of Z genes, linked genes, you should find twice as many reads in males because they have 2 copies to ZZ, and only compared to females that only have 1 Z. And so this is what you find. This is the type of scaffold data that we see that tells us that that scaffold is in a Z scaffold. Whereas when you find reads that are found in females, but pretty much inexistent in males, in males, then those are W-like scaffolds. you can classify them as being from the W. And then we have some scaffolds like this one that seems to catch the transition between the pseudoautosomal region and the Z-like, because it goes from having a very strong Z-like signal to the overlap of both like the autosomes right there at this position. So we did that. We have our putative scaffolds, and then we have also the we did some SNP analysis to try to discard Z genes that could have a W homolog still lingering around that might be expressed and have a confounding effect in our data. And we look at transcriptomic data. First, we look at this embryonic series at 5 different stages of development of the Apollonis spinifera turtle and a TSD counterpart, the painted turtle Chrysemys picta. And we look at the trunk, the adrenal, kidney gonad, and the gonad separated here for these later stages of development. This was something that we were doing for a transcriptomic analysis of sex determination in embryonic development, but we used those data to look at sex chromosome dosage compensation in Apolloni. So if you look in these graphs, we have expression, the ratio of male to female expression. If it is the same, that ratio should be male to female should have a ratio of expression of 1. When you use a log2 transform scale, that 1 becomes 0. The log of 1 is 0. So this will be the line of expression if males and females had an identical expression of these genes. And this is the density, so this is the abundance of these genes with different types of expression. In the reddish, you have autosomal genes, and in blue, you have, in the greenish, you have the Z single copy genes that are found in the sex chromosome. And they're single copies, so there's 1 gene of these in females and 2 copies in males. And so, if there was dosage compensation, you should see both distributions aligning right at 0 here, because the male and female ratio, the expression will be identical, so the ratio will be 1. When there's no dosage compensation, males will have double the expression because they have 2 copies of these C genes, and that's what we see here. But notice that at stage 19 and 22, these distributions start moving towards the left, trying like closer to the parity of male-female expression. That was for embryos incubated at 26 degrees, a colder temperature, and this one at 31, a warmer temperature. You see a little bit of compensation at these 3 stages, but it's definitely more marked at the 26 degrees. In contrast, the painted turtle that produces only males at 26, only females at 31 degrees, all genes, because it has TSD, all genes are autosomal. So we look at the exact same genes from Apolloni, so that are found in Chrysemys. So the homologous genes of the autosomal genes in Apolloni and the homologs of the sex chromosome genes in Chrysemys. And what we find is that for those 2 groups, the expression is centers around a male to female ratio of 1, so parity for both sexes. If we look at this in a different way, what we are seeing here is dosage compensation in embryos of the Apollonis spinifera turtles that is age, that changes by tissue, that changes by age, and that changes by temperature. So it is the first case of environmentally sensitive dosage sex chromosome dosage compensation that we know in eukaryotes. And this is coming up in the proceedings of the philosophical transactions of the Royal Society this very soon. This is another way of looking at that. We were trying to dissect how this is happening, and what we found is that here in the pinkish is the female data, in the bluish is male data, and we have different colors, different symbols. In circles is data from the colder temperature, triangles for the warmer temperature. In this side, the left, you have the Z chromosome data and the autosomal genes on the right panel. What you find is that the level of expression in the Y axis for the different stages of development in the x-axis here, for the females, they have very similar expression levels than the autosomal genes for each of these stages, right? These three first stages, the last two, the last two. So the females have an expression for the Z genes that is similar to the autosomal expression, even though they have only one Z chromosome. And that would tell us, there is dosage compensation in the females. But notice that the males have twice as much expression than the females for these Z-linked genes. So what seems to happen is that both sexes are ramping up the expression of the Z genes, particularly early in development. And in these later stages of development, we see the downregulation of the Z expression in males that is even accentuated further, more extremely at the colder temperature of 26 degrees. So it is a type 4 dosage compensation where you see first the ramping up of both, similar to C. elegans, ramping up of both of the Z expression in both sexes, and then down regulation of the Z expression in the males, and particularly at 26 degrees. We also look to see whether that's a pattern that existed in another way of looking at dosage compensation is to compare to see if the expression in the species with X chromosomes is the same as the proxy for ancestral expression in a TSD species. Our proxy for that is the painted turtle. So what you do is you calculate a ratio of ratios, the expression of the male Apolloni as a ratio compared to the same genes, but in the painted turtle. That divided by the expression of the females in Apolloni as a ratio of the expression in the painted turtle for that sex. So it's a ratio of ratios, and that gives us this RDC index that tell us whether there's a change from the ancestral expression or not. And we see pretty much the same pattern that we saw on the left graph here, that there is compensation that changes a little bit here from the no dosage compensation. We see these stages where there's a decrease towards compensation in the Apolloni turtle. And it is different than what was present in the painted turtle. We try to see whether this was done by, this compensation was happening by regulating globally the whole Z chromosome or gene by gene. And when we coded the compensation that we found in Apolloni by genes that had no compensation, a little bit, so partial or full compensation in these three colors. And what we have here is the scaffold along the Z chromosome, color-coded, and found reordered these genes for the two temperatures by the pattern of expression. And we've detected this group of genes that have little compensation early in development, these first stages of development, but then by the stage 22, they reach full compensation. And another group of genes that doesn't attain full compensation, but only partial. And this is accentuated at the colder temperature, 26 versus 31. Now, when we reorder these genes by the scaffold, so now the scaffolds are in the same color here, we see that these different patterns from group 1 and 2, and even this little group 3 that has no pattern discernible pattern there, we see that that you find different types of genes throughout along the chromosome. So this suggests that there's a gene by gene regulation of the dosage compensation in abalone turtles. And we look at other stages and ages, we find that in hatchling, so post-hatchling, in hatchling gonads, in liver, there's no dosage compensation. The Z genes have twice as much expression in males than in females, but autosomes show parity. But we see the same in the adult gonads. In the adult liver is the only one where we see dosage compensation. That seems to be full dosage compensation. Hatchling blood, no compensation. And we compare that with the painted turtle here and show that there's parity for painted turtles, parity here for the adult liver, but not for the other tissues. However, for the adults, we only had 1 male and 2 females, so this is tentative result. Now let me very briefly tell you about Glyptemis insculpta, the XY species that has this Y with only these bands that are male specific. For that turtle, we have expression data from hatchlings, gonad and liver of hatchlings, and this is the contrast with painted turtle, hatchlings, gonad and liver also. And we find, in this case, we have log expression here for each of these 4 columns. And we have the parity would be the autosomal for males and females in 2 different colors, female in the pinkish, pink color, and in green is males. They are similar for the autosomal genes of all of these. The pseudoautosomal region in the sex chromosomes of Glyptemis also have similar expression, males and females. The same is true in Chrysomis homologous genes, the corresponding genes. But in the Y chromosome, we have only been able to identify 8 genes that are single copy and have no homologue in the Y that is expressed. Much different from the abalone, perhaps because our genome assembly for Glyptemis is not as good. But irrespective, the point is here, I have the data individually for 8 genes instead of having a distribution of values like before. So if we look at this in the, one by one, we see parity between the sexes, same expression for several of these in the gonad or the liver. But what matters where I put the arrow here is to indicate that there was a change in expression compared to the expression of that gene in the same tissue in the painted turtle. So with respect to this, in the painted turtle here, this gene, the male expression moved to the right. So there was more expression in the male that brings that to parity. So this is indication of this gene being dosage compensated in the gonad of Glyptemis. Same for this gene in the gonad, same for this gene in the gonad. There's an increase in expression compared to the ancestral expression represented by the painted turtle, okay? Also, the same with this one. In fact, this one goes a little much, and there's much more overexpression, male bias in the liver of Glyptemis. So, but we are trying to, this could be, so there are several possibilities for this data. We only have 8 genes identified, perhaps because this is a young sex chromosome and there hasn't been degeneration in other genes to the point that they need to be dosage compensated. So they are not single genes, they are still expressed and they haven't changed much. The other possibility is that our genome assembly is not as good. So we are trying to improve that now and see if we can identify more genes in the Y and the X chromosomes of glyptomies. And this is compensation of the X. Just to clarify that. But the Y has not degenerated as much. And therefore, we only have 8 single copy genes as a reality, not because it's an artifact of a bad genome assembly. But we need to test that still. These are the same data, but now doing the ratio to look for differences with the ancestral expression in the painted turtle. And so here we have the log2 of that index, the RDC index. 0 will be a full dosage compensation here. And towards the bottom, female bias expression, towards the top, male bias. And for autosomal genes, there is full compensation. There's a parity here. But for the 8 genes, we see some that have full compensation, whereas in some other cases, we see male bias there. We see female bias in this gene, and in those genes, male bias. That's in the gonad, and in the liver, there's a couple here with full compensation, this one and that one, but not as much for the others. So what we have in terms of conclusions here is that I hope I was able to show you that the sex chromosomes in turtles evolve independently multiple times, that this evolution is tied to climate change events, that these ZW and XY chromosomes co-opted different autosomes to become sex chromosomes. The rate of evolution of those changes, the content of these chromosomes is different. I didn't touch into this, but we also have evidence that there's faster Z evolution, but slower X evolution in some of the sex chromosomes in turtles. And that we do see that dosage compensation in turtles. It happens to be thermosensitive in Apolloni, changes by tissue, changes by age, is full dosage compensation in females by bringing up the expression of both Zs in both sexes, and is partial in males once the male downregulate that in Z expression, the overexpression. Find it in some tissues, in some hatchlings, and only suggestive in adults, but it seems to be regulated gene by gene, not globally, and is partial. And it is different from the full dosage compensation found in Anolis carolinensis lizards that have a homologous sex chromosome system. So the XY of Anolis comes from the same autosome as the ZW of Apolloni. Yet in Apolloni, we see partial dosage compensation with this pattern that I show you, whereas in Anolis is full dosage compensation. In glyptomies, the sex chromosomes are young, and we see some evidence of gene by gene dosage compensation, but we don't have not enough data for very strong conclusions there. So we need more work to do still. So it's good to use these outgroups because it tells us also by using the painted turtle that the Apolloni was evolved dosage compensation by changing the expression from what was found in the TSD ancestor, whereas glyptomies is co-opting some of the expression that was present in the painted turtle. So there's a difference between those two, which is a closer relative of the painted turtle, this glyptomies one. And this environmental sex dosage compensation, we don't know how prevalent it is, if it's adaptive or not, what other phenotypic effects it might have. So there's more work to be done in the future. And with that, I will be happy to answer questions. Thank you.
Hi, Nicole. Thanks a lot. It was a beautiful talk, lots of information. I'm still trying.
Oop, I lost your sound.
Marta? Oh, so thank you very much for your lecture.
Yeah, my
Marta
connection,
returned.
you know,
Yeah.
like on TV, my connection fell, but it's like on CNN. So Dr. Gatto, he thanked for your presentation, and then he wrote, is it possible that past climate change events were crucial for the rise of GSD in turtles? And he completed, is there some evidence that the transition from TSD to GSD in some turtle lineages coincides with climate change events?
Yes, so we found from that work in 2011 that when we superimpose the global temperature with the timing of a split of lineages where sex determination took place, that split of those lineages coincided with peaks of global temperature or times of rapid warming or transitions from one extreme to another, like rising to lowering of temperature. So there is the possibility, this is, there was no statistical test, but just eyeballing that those transitions could have been aided. And you can imagine that if there's global warming and there's in most turtles, they produce females at warmer temperatures. So you can imagine that if in several of those events, the Earth was warming, TSD populations were feminizing rapidly, they could become male limited. So that if there's very few males, then all of a sudden there's selection for any female who has a mutation that produces males in their offspring, no matter what the temperature of the environment. And that would have all the ingredients for the evolution of an XY sex determining system, for example, or just a sex chromosome system. So there is that level of evidence, but that's as far as we go. What will happen in the future, we don't know. The turtles are imperiled by other factors, so it may not be just climate change like sea turtles, that we see some populations very feminized, but it may be that they die from habitat destruction or over exploitation, as occurs in our countries, in Brazil and Colombia, and everywhere.
Yeah, now
really.
I got curious about these genes that you found to be involved in sex determination. Do you know about their function? Because it seems that they are all related to climate somehow, with temperature.
So some of these genes, they are common genes in the vertebrate sex determination network. So we know their function from work in other model systems, in chicken, in mice, and so on. Some of them we know that are sort of what they do, that's well characterized. The most recent work in terms of genes in turtles, we know that there's also epigenetic mechanisms that take place, particularly for TSD. There's some temperature sensitive genes that are there's a histone acetylation that happens, for example, a trimethylated histone that attaches in the promoter of the MRT1, that sex determining gene in birds, and is male determining in many vertebrates. And there's an effect of temperature on another gene that causes the demethylation of that histone, and therefore impacts the transcription of the MRT1 by affecting the histone that is acetylated, that is methylated.
Interesting.
Yeah.
So the next A question is from Caroline Machado. She says that she's a fan and that
Thank
she
you.
loves your work and that it inspires her a lot. And she has been working on sea turtles.
All right.
So she asked the question. We had found that the Gata-8 microsatellite showed accumulations, mostly in microchromosomes. However, the absence of size heteromorphism between homologous chromosomes carrying the sites reinforced the condition of the absence of heteromorphic sex chromosome in this group. Gata is known to be in the promoter region of the DMRT1 gene. She's asking if that's the case, and what do you think about that?
I don't know. I couldn't tell you what's the characterization of the promoter regions in of the DMRT1. There's a student of mine who's doing one of her thesis chapters is looking at the evolution of promoters. So I will be able to tell you that in maybe a year or two once she finishes that. But there's microsatellites that are accumulated in many sex chromosomes, and there's good work from the Australian group of Arthur Georges now that have characterized this accumulation in multiple species, and that has been used also to characterize sex chromosomes just by that difference in accumulation in other turtles with GSD. Is a little bit tricky sometimes because there's, as I was talking to some groups like the group in Prague from Lucas Krakovil, that sometimes that can give you misleading signals. So one has to be cautious about that, of just using the macrosites, satellites, yeah.
Okay, thanks. Now, Diogo Melo says, thanks, Nicole, for the wonderful presentation. What are the perspectives to study sex chromosome evolution and function among turtles, considering the advances in the epigenomic field?
So I think that there's a lot to be learned from epigenetics. We've started working with methylones, and we did a little bit of work with the histone deposition also. And there's we have a paper when we were looking at the transcriptome in this embryonic series, and we concentrated one of the analyses on epigenetic genes. And this is from painted turtles and the Apolloni, the social turtles. And we found that a lot of the epigenetic machinery genes were responding to temperature, not only in the painted turtle, but also on the Apolloni turtle. And so there's and we're talking about genes involving methylation, in acetylation, in ubiquitination, you name it. So the machinery is being affected by responding to temperature. How that translates to phenotypic effects, we don't know yet. But there's a huge amount of work that could be done in that area. And with respect to the sex chromosomes, again, it's an open box, an open question, because we look at methylation in Apolloni, thinking that perhaps there could be dosage compensation by methylation. But when we compare methylation in cytogenetically by using antibodies against methylated cytosines, we don't see differences between males and females. We do see differences between microchromosomes and macrochromosomes, but not between the sexes. So that was didn't lead to something about dosage compensation, but I found the pattern still interesting, and it was different for different turtles. So there's a lot that could be learned in that area. It's completely frontiers field right now.
Right, and he also asked, do you believe that the transgenerational epigenetic marks would have a great contribution for the promiscuity of sex determination in the group?
Don't know. We don't know yet, for example, if in turtles, whether epigenetic marks are passed on from mother to offspring. So for instance, we have methylation data from hatchlings, and we have methylation data from stages 9, 15, and 22. And we do find differential methylation between the sexes in all four, and in the gonads in all four. What the next thing that we were planning to do was to look at differential methylation in adults and in mothers, and if we can have mothers and their offspring, then we can test that question, whether the pattern in the females is the same as found in the embryos, and whether there's erasing of the methylation marks and then reestablishment in the embryo, or whether something is passed on from mother to offspring. We don't know yet.
Okay, now I'm gonna pass myself in front of everybody and ask you a question. I'm curious because I never followed a lot of work on turtles, but I used to read a lot about sex chromosome determination in snakes that also have all this variation in different kinds of sex determination. So I would just want to, because I haven't been reading about it, if the information on snakes and lizards is also as advanced as the kind of information you presented on turtles.
So in lizards, there's good data on some groups, but lizards are so abundant, so specious, that we're starting to get more information. There's good detailed information from Anolis, from a couple other lizards, from Pogona, from the groups in Australia, and a couple of others. But there's a lot of transitions that are happening in lizards. We're talking about repeated evolution from TSD to GSD and back. So much more abundant transitions than in turtles. And not all of those are well characterized. Some are characterized by the simplest of methods, like PCR. Once you get one species that you know has XY and they found some markers, then they look for double the amount in one sex versus the other by PCR to try to screen large groups of species. And that has been useful because doing cytogenetics with all of them for a group that has 9,000 or 5,000, 9,000 species is just too much, right? So there's some groups that are studied in detail, some only superficially. In snakes, similar things. So the snakes all have GSD, and they were thought to all have ZW chromosomes. Now we know that yes, there are GSD. So far, there has not been no known system of temperature dependence in snakes, but that there's the boas and the pythons that have XY. They recently discovered. So that was very exciting, because that meant that a group of snakes that were thought to have a very homomorphic ZW system, it actually didn't have ZW at all, but had the XY. And which is also not very different, is not very heteromorphic, but that's interesting in itself too.
Yeah. But they didn't get two genes like you did with turtles.
No, there's only one. Well, there's only one paper from that compares several of these, including a boa, I believe, and some other snakes that did dosage compensation from 2013. That's from Vicoso. Yeah. But that's it.
Because, I have the impression that people start working a lot with the group and then they stop. So I worked with Jamie Grace too, and then at the time, she had people like ex-students and stuff doing a lot of work on lizards. And then it stopped completely. With snakes, there were some groups in Brazil that used to work on that on the 60s. And then they disappeared. So it's a pity.
It is.
other people also want to make questions. So I will be.
That's it.
So Ana Paula Moraes, she just thanked you for the amazing talk. And then Luciana Lorenzo, also thanks for such a great lecture. And that's a very interesting question. You mentioned a correlation between transition in sex determining system and changing diploid number.
Mhm.
Could you talk a little bit more about this?
Absolutely. So that was a study we published in 2011, so 10 years ago now, when we did phylogenetic analysis, because I noticed that when you look at different families of turtles, whenever there was a change in sex determination, the average number for the family was different. And then we did a species level analysis, proper analysis, phylogenetic analysis, and there is a correlation. So whenever there is in the branches of the tree of turtles where there's a change in sex determination, either TSD to GSD or the opposite, there is 20-fold higher rate of evolution of chromosome number. So somehow the genomes are reshuffling, the chromosomes reshuffle at a rate that is 20 times higher when there is a change in sex determination. Now that could be cause or a consequence, right?
Yes.
So we try to do when we did the analysis to look for the directionality. So we see whether one trait changed first and then the other, or the opposite, and there was no difference, as if they in geological time happened pretty much at the same time. So very, very close together. That still could be that one causes the other, or the other way around. And we propose a couple of hypotheses. One is that it could be that an ecological factor, say climate change, which we did find was correlating with the split of these lineages where the change determination was occurring. Perhaps there's a change in climate, there's a favor, there's a natural selection to change the sex determination. And if there's a mechanism that is already involved in sex determination that changes during that transition, and by changing, it makes the genome unstable, then that may cause the chromosomal rearrangements that if there are fusion or fission, then it increases or decreases diploid number. And there was no directionality there. It was just rate of change was 20 times higher, but not necessarily always increasing diploid number or always decreasing. It could go either way. The other possibility is that for whatever reason, the fusion and fission of chromosomes happen first. And once that happens, they may change the relative position of genes that are important in sex determination. So say you have a chromosome that contains a block of genes that are important for sexual development. Now you have a fusion, and these were regulated in cis. Now they're into chromosomes, and now they are in trans. And that could change. If they happen to be important for sex determination, perhaps that then causes a transition in the sex determining system. Perhaps it's sometimes one way, sometimes the other. We don't know. So we're trying to explore that. One way is to reconstruct the turtle karyotype, and we have done a lot of work, but we still need to finish that up. Look also with the genome assemblies that we have for the genes around the break points of chromosomes, and see if they are involved in sex determination. And we have the bioinformatic analysis from the genomes, and also back clones that we can use to try to place these genes and see if there's an association between those changes in chromosomal location for genes that associate with fusion fission and the changes in sex determination. But yeah, that's where we are at the moment.
So I think we are done with questions. Thank you very much.
Thank you.
It was a lovely talk. It was very beautiful.
Thank you.
I think Daniel wants to say something.
Yes. Thank you so much for your presence here, Nicole. We have a good time for together you here.
Yeah.
A lot of people, thanks for your presentation and very interesting to reading about your papers.
Thanks.
And was a really pleasure listening to you today. Nós finalizamos aqui a nossa live de hoje e convidamos a todos para o nosso próximo encontro, que ocorrerá no dia 9/6 de 2021, com o tema Mapeamento óptico para a investigação e caracterização de rearranjos estruturais no genoma. Quem vai ministrar essa palestra é a doutora Cláudia Carvalho Fonseca, da Pacific Northwest Research Institute, e a doutora Juliana Forte Mazeu de Araújo, da Universidade de Brasília. Nós agradecemos a presença de todos e nós vamos finalizando nossa transmissão aqui. We are finished your broadcast in this channel. Thank you and have a nice day.
Very welcome. Thank you.
Nicole, stay there.